Although compact bone appears very dense and smooth with the naked eye, microscopic evaluation reveals it is riddled with canals and passageways which serve as conduits for blood vessels and nerves. The structural unit of compact bone is called the osteon or Haversian system. An osteon is an elongated cylinder oriented parallel to the long axis of the bone, analogous to tiny weight-bearing pillars. An osteon is a group of hollow tubes of bone matrix, one placed outside the next, like the growing rings of a tree trunk. The hollow tubes are called lamellae. All collagen fibers in a single lamella run in the same direction. However, the collagen fibers in the adjacent lamellae run in the opposite direction. This arrangement allows for the resistance to torsion stresses. Running through the core of each osteon is a central or Haversian canal. The Haversian canal contains small blood vessels and nerve fibers that serve the needs of the osteon's cells. Perforating or Volkmann's canals lie at right angles to the long axis of the bone and connect vascular and nerve supplies of the periosteum to those in the central canals and medullary cavity. Osteocytes occupy small cavities known as lacunae at the junctions of the lamellae. Hair-like canals called the canaliculi connect the lacunae to each other and to the central canal. Between the osteons are incomplete lamellae called interstitial lamellae. These fill the gaps between forming osteons or are remnants of osteons that have been cut through by bone remodeling. Circumferential lamellae are located just deep to the periosteum and extend around the entire circumference of the shaft. So once again, the principle of complementarity is evident in the structure of bone. Clearly, the structure of bone denotes its function to provide support and protection. Its hard matrix provides for strength. Instead of being made up of a single continuous structure, it is made up of several components that allow for summation of strength and a diversity of protections. The bundled nature of the osteons allow for a summation of strength. Not only is the bone matrix hard, it is reinforced by collagen fibers in the lamellae, and not only are the collagen fibers present, they are arranged in patterns that help the bone resist torsion forces. Finally, bone also exists as spongy bone. The trabeculae of spongy bone are precisely arranged along the lines of stress. They serve to provide strength without adding excessive weight to the bone. The chemical composition of the bone matrix consists of both inorganic and organic components. The inorganic components make up 65% of the mass of bone and consist of hydroxyapatites or mineral salts, largely containing calcium phosphates. The calcium salts lie in and around collagen fibers in the extracellular matrix and account for bone's hardness. The organic components include the cells, which are the osteoblasts, osteocytes, and osteoclasts, and osteoid, the organic part of the matrix. About 1/3 of the bone matrix is known as osteoid. Osteoid is the organic component of the matrix. Osteoid includes proteoglycans, glycoproteins, and collagen fibers, all produced by osteoblasts. The cells of bone tissue include osteogenic stem cells that differentiate into osteoblasts. Osteoblasts produce the fibers and the extracellular matrix of bone tissue. Osteoblasts are non-mitotic cells and also participate in the bone mineralization process. Some osteoblasts mature into osteocytes and serve as strain sensors and exert chemical influence over osteoblasts and osteoclasts. Osteoclasts are large, multinucleated cells responsible for the degradation of bone tissue to facilitate bone remodeling, blood calcium levels, acid-base balance, and other important physiological processes. Because bone is connective tissue, it has its origin in mesenchyme. Mesenchymal cells ultimately differentiate into the specialized cells of bone tissue. At 6 weeks, the skeleton of the human embryo is constructed entirely of fibrous membranes and hyaline cartilage. When bone develops from a fibrous membrane, the process is called intramembranous ossification, and the bone is called membrane bone. All flat bones derive from the intramembranous process. Bone formation that occurs by replacing hyaline cartilage is called endochondral ossification, and the resulting bone is called cartilage or endochondral bone. After 6 weeks, the mesenchymal cells begin to differentiate into hyaline cartilage cells and form a hyaline cartilage model of the developing bone. A bone collar forms around the diaphysis of the hyaline cartilage model. As the bone collar forms externally, chondrocytes within the shaft hypertrophy and signal the surrounding cartilage matrix to calcify, forming the primary ossification center. Because calcified cartilage matrix is impermeable to diffusing nutrients, the chondrocytes die and the matrix they maintain begins to deteriorate and cavities are formed. Cartilage elsewhere remains healthy and continues to grow, causing the entire cartilage model to elongate. In the 3rd month of development, the forming cavities are invaded by a collection of elements called periosteal bud. The bud contains a nutrient artery, veins and lymphatics, nerve fibers, red marrow elements, osteoblasts, and osteoclasts. The entering osteoblasts secrete osteoid around the remaining fragments of the hyaline cartilage, forming a bone-covered cartilage trabeculae, the earliest form of spongy bone in a developing long bone. As the primary ossification center enlarges, spreading proximally and distally, osteoclasts break down the newly formed spongy bone and open up a medullary cavity in the center of the shaft. The formation of this cavity is the final step in the ossification of the shaft. Throughout the fetal period, the rapidly growing epiphyses consist only of cartilage, and the hyaline cartilage model continues to elongate by division of viable cartilage cells at the epiphysis. Since cartilage is calcifying, breaking down, and being replaced by bony spicules on the epiphyseal surface facing the medullary cavity, ossification chases cartilage formation along the length of the shaft. Shortly before or after birth, a secondary ossification centers appear in one or both epiphyses, and the epiphyses gain bony tissue. The cartilage in the center of the epiphysis calcifies and deteriorates, opening up cavities that allow entry of a periosteal bud. Then bone matrix is secreted around the remaining cartilage fragments by the entering osteoblasts. In secondary ossification, the spongy bone in the interior is retained and the medullary canal is not formed. When secondary ossification is complete, hyaline cartilage remains only at the epiphyseal surfaces as articular cartilage and at the junction of the diaphysis and the epiphysis, where it forms the epiphyseal plate or the growth plates.